Thank you very much, and we continue our series of lectures, and next will be Dr. Xianmim Zeng. She is an assistant professor and director of the North Bay CARM Shared Research Laboratory for Stem Cells and Aging at the Buck Institute for AIDS Research. Her presentation will be on human embryonic stem cells, potential therapeutic applications for neurodegenerative diseases. Please welcome Dr. Zeng.
Good morning, everyone, or maybe good afternoon. So it's a pleasure to be here, and I want to thank the Dominican Republic, Dominican University and the Zero Breast Cancer. It's very bright, the light here. Sorry, it's a pleasure here. So I think what I want to, you see the picture here, that's actually Buck Institute, and I guess most of the audience sitting here, maybe from the Marin County, you probably have visited our facility or research center there. But here is the building where we, it's actually an IMP design, so we have the lab and administrative part. So I think I'm not going to go over the stem cells and definition, and as all the other panelists that have done a lot of that job, and which is great. So I'm going to focus more on the therapeutic potentials. I'm going to majorly illustrate this in an age-related disease. It's a neurodegenerative disease called Parkinson's disease. Can I go to the next slide?
Keep going.
Okay. So the type of the stem cells we have doing research at Buck Institute is majorly focused on human embryonic stem cells. And I just list a couple of four of them, the therapeutic potential for this type of stem cells. Mostly we are focused on developing therapeutic strategies for treatment of currently non-treatable or non-curable neurodegenerative disease. And we also use the genetic modified cells to try to screen drugs that can probably, maybe in the future, be used for treat disease like Alzheimer's, Parkinson, or Huntington. They have the neuroprotective effects. And also, you can use these cells actually to study the aging process. You probably have heard a lot about the potential of stem cells. They can keep on dividing, means actually they are not dying. So these cells are not aging. So using these cells to study things that do not undergo aging, you probably can find some clues for why we are age, okay? And finally, you can use these cells as a disease model you can study in vitro, probably find the pathology of the disease or therapeutic potentials. Next slide. So skip this. Please skip this. So I just want to show you some of the embryonic stem cells we culture at Buck Institute. And actually, I originally started human embryonic stem cells work at the NIH. So we have worked with this human embryonic stem cells for many years. And we first, of course, can grow these cells in the feeders, as you can see, the mouse feeders, and then we have improved the technology to grow them in a feeder-free condition. But these cells are grown on a media that has been conditioned with the feeders. And finally, we move to this. We can grow the cells in a totally chemical-defined media. So if you want to use stem cells as therapy for transplantation, you will have to grow the cells, for example, without contamination with the feeders or contaminate with the animal proteins. So this is essential. You should be able to do culture or prepare your cells in a media that is not contaminated with the animal proteins or serums. So this is just a picture to show you a nice colorful picture that distinguishes the stem cells, this label with the red, from the mouse feeders. Next slide. So now I'm going to talk about therapeutic strategies for treatment of neurodegenerative disorder. Again, I want to start with Parkinson's disease. Next. So what happened in the Parkinson's disease brain, you probably have heard, what happened is in the midbrain in a small spot called substantia nigra. And then they have a type of neurons called dopaminergic neurons. Somehow, it can be genetic or environmentally. It also can be just randomly. They die. So, next slide. And you probably have heard there are a lot of treatments for Parkinson's disease, especially in the earlier stage of the disease. What you try to do is actually you try to supplement dopamine in one way or another that can try to replace the dopamine cell, producing cells that has been degenerated or killed. Again, there are also people try to use surgery procedures, like you try to stimulate your brain, but this is not, it's as some kind of successful, but as the disease progresses, it's not really cure the disease. And also small molecules try to prolong the survival of the dopaminergic neurons. So these all treatments are really do not repair or replace the neurons that are lost there. Next. So in the earlier 80s, actually, people have proposed a new strategy to treat uncurable neurodegenerative disease. That just assume if you can put new neurons into the brain, into that part, and if these neurons can form the network with the existing networks, the neuronal networks in your brain, and perform the function. Oh, thank you. So, okay, can you hear me?
Yeah.
Okay. So if you use the newborn neurons to replace the lost neurons in the substantia nigra or in the midbrain, so you may find a cure for this disease if this is successful. So one problem for this is neurons that can be used for transplantations, they must be immature neurons. You cannot get any mature neurons from your brain and put it there. They're not going to divide because neurons are actually post-mitotic, means they're not dividing anymore. Once you get your neurons, since you were born or in the fetal development, you probably just get all those neurons through your lifespan. So you should use immature neurons. So far, the only sources or reliable sources that you can get these neurons are actually from a fetus. In the 80s and the 90s, there were actually several clinical trials using fetal dopaminergic neuron transplantation for Parkinson's disease. One is conducted by Kurt Fried at Colorado University. So one problem is that one Parkinson's disease patient, for one single transplantation, you will need about 6 to 10 fetus of about 12 weeks after fertilization. So that's a very big challenge. And also, it's depend on the fetus available. You are very difficult to prepare the cells. You know, you cannot replicate them exactly the same. So alternative, since the derivation of human embryonic stem cells in 1998, so there was a tremendous interest and a lot of investments to try to generate dopamine neurons from human embryonic stem cells. And if this can be successful, then you probably have a very promising unlimited source for dopaminergic neurons. So why I'm talking about Parkinson's disease as an ideal candidate for cell therapy, I must say there are other diseases like spinal cord injury, type 1 diabetes, and there are also ideal candidates for cell therapy. But one thing good about the Parkinson's disease for ideal candidate is because, you know, the damage in the brain is relatively focal, so they are only involve a few type of neuron populations, mostly the dopaminergic neurons. And you can assume that, or you can maybe, this may indicate that a relative small numbers of GN cells could restore the function, okay? So another advantage of Parkinson's disease is actually we have already developed models, including rodents and the non-primate models, and those are ready for testing. And unlike disease like Alzheimer's, it's really difficult for doing transplantation in my view, because you don't even know which models you can be used for transplantation. So also there are data from the human trials, though they are inconsistent. But they do provide some clues, like which part of brain you have to do the injection, and how many cells you probably need, and how often you need to inject. So all those you can. There are some groundwork has already been done for Parkinson's disease. So now that's the basis. So I want to show you some of the neurons we have been generated from our lab, just to show you this is an immunostaining that you can actually use different markers, as the previous speakers have been talking about, that you can label. You like Dr. Pera just said, for a neurologist, we probably say there are 200 types of different neurons. So you don't want to transplant all different types of neurons into Parkinson's disease patient. So you have to specify them. So what the color you see here, the label, when they are co-labeled with green and red, they turn yellow. And those are actually, the red marker is label all the neurons, and the green markers specifically dopaminergic neurons. So I just want to show you one of the slides of the transplantation. This is, we have done the transplantation into the rat Parkinson's disease brain. Just want to show you that the cells does survive the transplantation. This is a picture took after 8 weeks of transplantation. And the behavior data also indicate that they can relieve Parkinson's syndrome symptoms, at least in rats. So I think this is a nice picture, actually, and this is the one I was giving a presentation at the NIH in 2005, I must say, 3 years old. And that time we were asked, like, how long and how, use cell therapy. I just say this is an estimation, or is an optimized estimation. So just to emphasize that you need to use the animal models before you go for the human trials. So this is our drawing at that time. We think about you need to have that much animals to get sufficient data to put into a human phase 1 trial. So if you don't do animal studies, you all use human subjects. That's going to take long. And a lot of the persons will probably didn't show any therapeutic benefits. So the next couple of slides, I just want to show you, you probably heard you can use this to use embryonic stem cells to screen drugs. But how do you do it in a lab? Or maybe later in a large scale, you can do the high throughput screening and work this together with the pharmaceutical companies. So I want to, again, illustrate this principle by one of the drugs called MPTP that's actually killed the dopamine neurons. And this is the drug, actually, Dr. Bill Lunston at the Parkinson Institute now. And he first found in the 80s, they found a group of California young drug addicts. They developed the symptom of Parkinson's disease, which is very strange because normally you connect the Parkinson's disease to old people, not young people. So what they found is this group of drug addicts, they took a drug contain a component called MPTP, which is complicated, but this is the active metabolic of MPTP, MPP plus core, that's actually selectively kill the dopamine neurons. And since then, people have used this drug to, to make animal models for Parkinson's disease. So when we derive dopaminergic neurons from human embryonic stem cells, there was a race in like 2004. There are 3 labs. Everyone tried to get published first. So we end up published almost the same time, all the 3 labs. And at that moment, we decide we want to see whether these cells can be used really like behavior, like the neurons isolated from the brain, and not like some of the cell lines that you immortalize them by oncogenes. So we want to test whether these cells respond to toxic agents and neuroprotective agents as they're supposed to. So I don't think I need to explain you, even from the lay audience, you can see the difference between after you add the drug for example, like 24 hours. These dopaminergic neurons are really, they're dying. And also they lost all the processes. So, and then, importantly, can we find any drugs to prevent or to help the dopaminergic neurons survive? So this is a drug called GDNF. It's a neurotrophic factor that has been used in the clinical trials in the 90s for Parkinson's disease patients. So we try to add this drug into the cells that has been exposed to MPTP to see if they can recover. This is all done in vitro. And as you can see here, you add the drug and you have the increase of your dopaminergic neuron survives. So that just say 2 of the big potentials for human embryonic stem cells. Well, you want to use it as an potential cell source for cell replacement therapy and also for drug screening. But there are a lot of things I think actually people are not, at least not in the general public, people have been focused on therapy, but there are a lot of potentials. For example, to study the developmental biology like Dr. Pera just talked about. And there are also something I want to tell you. We can use embryonic stem cells to study the aging and aging-related biologic process. So what you have learned today, hopefully, you have learned normal cells, if you take your cells from adult, they will divide a limited time, depend on how old you are and which type of the tissues the cells are coming from. But eventually, if you take a young fibroblast from the infant, you get them like 50 divisions. But essentially, they will all undergo senescence. They are still alive, but they are not dividing anymore. Typical example is the neurons. You have it, like for 80 years, if you live 80 years. So, but they're not dividing anymore. They still perform their functions. And this phenomenon is called senescence. And this is a word just like aging. So, and you also learn human embryonic stem cells. They can grow in culture indefinitely. That means actually they can bypass this senescence. How they do it? There must be many mechanisms how they do it. But embryonic stem cells, as they differentiate into somatic cells, then the ability to bypass this senescence is lost. So you can imagine if you study the process of embryonic stem cells differentiation, and how they undergo from undying cells to aging cells. Study those mechanisms. Maybe it will give you some clues why we are aging. Okay? So I just want to point out some of the process, aging-related processes, which all this has been done at the Buck Institutes. So of course you think about senescence and something called telomere that is also involved in the aging and how you repair your genome and how the cells, some of the cells, you have a mechanism for programmed cell death. So that means actually you do not get cancer. Some of the cells, they are programmed to die. And then you have some of the logic process is when the cell divide, they have the cell cycle. You look at those checkpoints, and all these are aging-related process, and you can use human embryonic stem cells to study this process. And then hopefully one day it will give you some clue why these cells are not aging and why are we aging, what we can do to prevent aging. As you probably heard last year's Nobel Prize winner, they are doing the work to genetic modify mouse embryonic stem cells, and those are really revolutionized the mammalian genome studies. So what a lot of people now are trying to do is to do this gene targeting, monitoring disease in human embryonic stem cells. Some of the disease like Huntington disease and ALS, you can just, do some of the point mutations or just do the knock-in. And if you get such lines, then you can, once you differentiate them to neurons or towards some specific pathways, you can study the disease, how they progress in vitro in a petri dish, and hopefully find some drugs to prevent. So this is the last slide. What we have done in our lab, we have tried to create a genetic modified cell lines. This is actually a gene targeting by homologous recombination, which you see is actually you made some specific change in the genome, and then this is just a gene specifically controlled the differentiation toward motor neurons and oligodendrocytes that would be very useful for disease like ALS or spinal cord injury and other motor neuron diseases. So I think I'll just end with here, the last slides, please. So, and I want to acknowledge the people at my lab work very hard. And some of the pictures are from Mahendra's lab and also from my previous lab at the National Institute on Drug Abuse. And the last slide you show is actually coming from Mario Capacchi. They made the construct for us. And I'm very grateful to the CIM for the funding, and also we have fundings from the NIH and from the Larry Hilbrun Foundation. Thank you.